Hello everyone, my name is Amirali Aghazadeh from University of California, Berkeley. Today I'm going to present a joint work with Orhan Ochal and Kanan Ramchandran on CRISPRLand, interpretable large-scale inference of DNA repair landscape based on a spectral approach. CRISPR-Cas9 is a technology recently developed by scientists at the University of California, Berkeley and Broad Institute around 2012. It appeared on the cover of several magazines at a time. CRISPR enables a simple and affordable way to manipulate and edit the DNA. It has completely changed the face of genome engineering. In particular, CRISPR has enabled us to better understand the DNA repair mechanism, which is an important research area in molecular biology. Now, you can think about CRISPR as a pair of tiny molecular scissors, which can create a double-stranded break at precise locations on the genome. Double-stranded breaks are detrimental to the integrity of the cell. So as a result, some repair mechanisms have been evolved, which joins the flanking ends of the broken sequence. In this talk, we focus on non-homologous end joining and macrohomology-mediated end joining, two repair mechanisms that repair the DNA in absence of a template sequence. These mechanisms have much higher edit efficiency compared to homology-directed repairs. However, their outcome is less deterministic. So as a result of the break, some short insertions and deletions happens in the vicinity of the cut site. In this example, a 6 base pair deletion is the most frequent outcome, but there might be other outcome as well. For example, a single base deletion or an insertion of letter G. So we can summarize this into a table where I'm showing all the possible insertion deletions in the first column and the fraction of the cells that gets each of these indel types in the last column. It has been shown around 2016 that the DNA repair outcomes is non-random, is actually a function of the DNA sequence around the cut. New algorithms have been developed to predict the DNA repair outcome from the DNA sequence. And these algorithms are pretty useful for many biological studies. For example, for a gene knockout study, one can use these algorithms to evaluate the efficiency of gene knockout by looking at the predicted value of fraction of cells that will get a 0 frame shift. So in this case, we have used the Indelphi software at this particular location of the genome, and Indelphi predicts that about 34% of the cell will get a 0 frame shift. And probably the most popular algorithms to predict the DNA repair today are Indelphi that we talked about, and Forecast and Sprout. So these algorithms are all developed around 2018 and 2019. And they are trained on CRISPR data from experiments on certain cell types, as you can see in this table, and they achieve reasonable accuracy in predicting key outcomes, such as frame shift percentage. However, challenges remain in understanding the DNA repair mechanism. And this is particularly because while these models are very good predictive tools, they are not fully interpretable. It's not clear what features in the input space, in the input DNA sequence correspond to which outcome. Secondly, it's not clear how many training samples are really efficient to reliably train these algorithms. Researchers typically resort to data from the experiments that they have obtained in order to train these algorithms. Thirdly, there are some scaling problems in the inference time due to an exponential growth in the problem size. But in this talk, I'm going to talk about our new spectral framework called CRISPRLand, which complements the current models in order to address these issues using ideas from signal processing, coding, and learning theory. So CRISPRLand analyzes the DNA repair landscape in the spectral domain. But the question is, what do we really mean by a DNA repair landscape? So let's take the example from the previous slides where we were doing a CRISPR cut experiments on a certain location on the genome. And we know that the Indelphi software has predicted the output of 0 frame shifts to be about 34%. Now, what we can do is that we can represent the DNA sequence using the string of +1 and -1 using this encoding table. Other encoding policies can also be used, but for this talk, I'll stick to this encoding policy. Now we can think about predicting the percentage of zero frame shifts as finding the function f defined over this binary string. Now let's take a window around the cut size of length 4 and define binary variables x minus 2 to x2 to represent these 4 locations. Now, let's fix the binary string outside this window, and let's start changing the binary patterns inside the window. And in particular, let's create all the combinatorial patterns over the binary variables. And for each pattern, let's construct this CRISPR experiments where we are cutting the sequence in the middle and find the zero frame shift value for that experiment and put it in the table on the right. So what we really call a CRISPR landscape or a DNA repair landscape is the vector on the right in this table. We can construct similar landscape for other DNA outcomes. For example, the fraction of cells that get a single nucleotide insertion, which we have shown in the paper, but I'm going to stick to zero frame shift as my running example in this talk. Function f in this slide is called a pseudo-Boolean function in mathematics. It's Boolean because the input is a binary variable, a high-dimensional binary variable, and it's a pseudo-Boolean because the output is not binary, it's actually a real value, for example, 34%. So it turns out that we can write down an arbitrary pseudo-Boolean function uniquely as a polynomial whose coefficients are the Fourier transform of the landscape, which is also known as the Walsh-Hadamard transform for binary vectors. So in this case, I have taken the y vectors in the table and represent y as a linear sum of the columns in matrix A, which is called the Walsh-Hadamard transform. So alpha is the Walsh-Hadamard transform of y. So what you can see is that the values in alpha uniquely corresponds to the monomial in the representation of the pseudo-Boolean function f. So our key observation in this work is that the DNA repair landscape is sparse in WHT. In other words, we can represent f as only a few number of monomials. In particular, out of 16 variables in alpha, most of them are 0 and only 4 are non-zero. Now, let's take a look at the same landscape when I use a larger window around the cut side. For example, in this case, I'm looking at the window size of 20. So I represent these variables as x minus 10 to x plus 10. Now the dimension of the problem grows as well. So the new dimension will be 2 to the power 20, which is about 1,000,000. So what I do is that I will query the in Delphi software 2 to the power 20 times, which takes about 2 days of simulations on my laptop. I construct the DNA repair landscape and find a Fourier transform. And this is the Fourier transform of the landscape. So it's interesting that the resulting landscape is very sparse in WHT. In fact, only 400 coefficients explains more than 95% of the energy in the landscape. To better visualize these coefficients, I zoom into the first 10,000 coefficients and display the monomials each coefficient corresponds to. For example, the highest coefficient corresponds to x minus 2, next one is x2. I also plot the top 100 WHT coefficients in this table, where each row corresponds to one monomial here. And the yellow dots correspond to the active variable in each of the monomials. So if I zoom in into the first 5 rows of this matrix, what we can see is that the top 2 monomials corresponds to x minus 2 and x2. Now let's think about how we can interpret these monomials. So let's focus on x minus 2. Realize the nucleotide that starts with +1 in this encoding table are T and G. So, and also realize that the coefficients of x minus 2 in this plot on the left is negative. It's particularly is -3. Therefore, This monomial x minus 2 is saying that the presence of a T or G and left to the cut side will deplete zero frame shifts. So you can also interpret the next monomial x2 the same way. In particular, you should realize that the only nucleotide that end with a plus ones are C and G, and the coefficients of x2 in the plot on the left is positive. Therefore, the presence of G or C at the right to the cut side will enrich for frame shifts. So overall, you can see that many of the coefficients, most of the coefficients in this table are centered around the cut side. So the interactions are right around the cut side. Now, if we zoom into the other block, the four monomials here reveal higher order interactions that try to capture macrohomologies, which are repeats of length 4 around the cut with 2 base pair in between. So we already know from biology that these sort of repeats were encouraged for a deletion as a result of the cut, a deletion of length 6 in this case. So overall, what we can see that CRISPRLand provides a platform that one can easily reveal the features in the black box predictive model by inspecting the output without even looking at what's going on inside the black box. So it's kind of agnostic to the type of predictive model. So we used CRISPRLand to compare the DNA repair landscape in the WHG domain across 3 different cell types, U2OS, MESC, and HCT. Surprisingly, what we found is that the location of the WHG coefficients were fairly consistent across cell types. And really the differences are on the relative value of the coefficients rather than the locations, probably due to the shared mechanistic behavior of repair across cell types. New research has to be done in this front, but the observation has been really interesting. But what if we want to look at a larger window size around the cut? And really the question is, what window size we should really look at? And in order to address this question, we have the plot on the left. So the plot on the left tells us that one needs to look at a window size of at least 40 in order to reduce the effect from the sequence outside the window to below 20% in the worst case. But if you look at on the right side, you realize that the dimension of the problem grows exponentially with n. In particular, for n equal to 40, you will need to query the Delphi software 10 to the power 12 times. It takes about 5,000 years to do this inference. So even using parallelization, it's impossible to get all the samples in silico, let alone doing this in the experiment. Therefore, there's a need for algorithms that have sublinear sample and time complexity, the ones that grow logarithmically with p rather than linear. Fortunately, fast and sample efficient algorithms have been developed using ideas from signal processing and coding theory, which can learn the landscape F in sublinear time and sample complexity, provided that F is sparse in the spectral domain. And this is something that we have already seen it happens in the CRISPR DNA landscape. So using these algorithms, we can cut the number of required samples to construct the DNA repair landscape from 10 to the power 12 to millions, which means that the time to collect the sample reduces from 5,000 years to only a week. So it's a significant reduction in terms of number of times you need in order to reconstruct the whole landscape. So we use the modified version of our sublinear algorithm to learn the landscape with a window size of length 40, using only a few million samples rather than 10 to the power 12 number of samples from the Delphi softwares. So these samples are designed based on some particular patterns coming from coding theory. We evaluated the performance of the algorithm in predicting the landscape on 300,000 random unseen CRISPR cuts. The prediction accuracies were pretty good on all the DNA repair landscapes that we tried. And in particular, we are showing two of the landscape in this case, which are insertion percentage and zero frame shift percentage that we have talked about. So in summary, we talked about CRISPRLand, a spectral framework that can help improve and enhance DNA repair models in terms of interpretation, reliability, and scalability. The mathematical framework behind CRISPRLand can be used for other landscapes, such as proteins, for protein design problems, which we are currently exploring. With this, I want to thank you for listening to my talk. And I want to encourage you to please check our GitHub for all the codes in order to reproduce the results in this work. And I want to open the stage for any questions. Thanks a lot.